I will ask you for this next part, we're going to move to chapter number 4 and start talking about how neurons actually work. So now we'll actually get into, as opposed to just kind of information sharing, how does an action potential work? An action potential is the basic mechanism that allows information to come into your body when cells in your rods and cones transform that light energy into electrochemical energy. That energy and those processes go all the way up to your brain. One of the key processes that enables all of this to occur is called an action potential. So we're going to go through in some detail here to understand what an action potential is, because we're going to be applying this when we learn other types of information. All of this information in the unit now, if you notice Roman numeral 4, is called electrophysiology. How the electricity of the process of action potential works. So the field of electrophysiology, we study how electricity works up in your neurons, up in your brain. The book has a lot of detail to this. I have less. You can go with me if you wish. If you want to have more, you can. Page 111 talks about the start of this. You can actually go into neurons and measure the difference inside a neuron. You can see the figure there, 4.2, versus out of the neurons. And it actually turns out that there's some differences during this first part of before anything happens to the neuron. Before anything happens to the neuron, it's considered to be in its resting state. The resting state is before any electrical stimulation comes through. So before you're asked to look at the 100 plus 25, the resting state before any activity comes through. We know that during the resting state, everything is not in complete balance in the neuron. And if you look at the figure there, even 4.1, you can actually see there's a layer separating the inside versus the outside of the neuron. It is called the lipid bilayer. It's a bilayer. You can see figure 4.1 there. If you click it on the link I've put in your handout, you can see another example of a lipid bilayer. During the resting state, we know a couple of key things. We know, number one, that there are ions both inside and outside the neuron. And it turns out that there is more sodium, Na+, on the outside of the neuron, and there is more potassium on the inside of the neuron. So those are two things that we know. There's more sodium on the outside of the neuron, there's more potassium on the inside of the neuron. Now, due to what we call diffusion, and it looks like the book talks about this, what normally happens is a little bit of sodium will start leaking in, and a little bit of potassium will start leaking out. But there's something that prevents that from really happening well, and that is called the sodium-potassium pump. Get the pump. Page 113 talks about the pump. The idea of the pump is the pump keeps the sodium outside and the potassium inside. So during the resting state, we know there's more sodium on the outside, there's more potassium on the inside, and if you look at figure 4.2, the other thing we can tell is if we look at the charge, it turns out inside the cell, there's a charge of about -70 millivolts. The inside of the neuron is -70 compared to outside of the neuron. So the three things we know during the resting state: there's more sodium on the outside, there's more potassium on the inside, and the charge is -70 millivolts. The sodium-potassium pump keeps that balance in place, and this charge, this -70, is called the resting membrane potential. Those ions are sodium and potassium. I know the book mentions other ions as well, but I don't know. I think we got enough here. There is a little diffusion happening. If you look at figure 4.1, you can see those little channel proteins, but the pump kind of keeps that balance in place. If you click it on the resting state video, you'll see a little more detail for this. This might help explain it to you. The book has even more details than this. I don't need you to know that. I need you to know what I've shared with you. So that's the local structural events. Now we got to move on to the next part of this. So how does an action potential begin? I wish I had a little picture here, but let me give this my best shot here. If you remember the three parts of the neuron, you remember what actually receives the electricity? If you go back to that picture, I think it was around page 39 or so. Let's see if we can find that in the book. Page 39. I think they showed a picture of a neuron. Did they not? There it is. The dendrites collect electricity, and so in order to start what's called an action potential, what basically occurs is when you see something, if you're asked what's 100 plus 25, when that light energy is changed into electricity energy in your retina, the dendrites are the first cells that actually collect that energy. In order to have an action potential, dendrites need to collect 15 millivolts of positive energy. Instead of positive energy, we sometimes use the term depolarization. So dendrites need to collect 15 millivolts of positive energy. So you can see on page 39 there are a bunch of dendrites there. The idea is if you have 3 dendrites and they each collect 5 millivolts of depolarization, positive energy, then that will all summate in the cell body. And if it's 5, 5, and 5, they'll summate in the cell body, and if they make it to 15, then that neuron will have an action potential. If it's 5, 5, and 4, then you won't get an action potential, because an action potential works on what we call an all or none principle. Either you have an action potential or you don't. So 15 millivolts of depolarization have to be collected from the dendrites. It has to summate at the cell body, and if it summates at the cell body, you will get an action potential. I hope that's somewhat clear. I hope the video that you watch might be able to explain that a little bit more. It says here on page 114, yes, you need 15 millivolts of depolarization. If you get that, you have reached a threshold. Now think of what the neuron will be if it collects 15 millivolts. If it starts at negative 70, it needs to get to a state of negative 55. And if it gets to a state of negative 55, then the events of an action potential will occur. So I hope that's clear. This is going to be a lot of detail here, so watch your videos here. Maybe it'll explain it better. Maybe you'll listen to my words a couple of times. So that's how an action potential begins. Now we have C and D on my outline here, a couple of other things that happen as well. We're going to talk about local structural events, and we're going to talk about signal transmission. So local structural events. What basically happens is if you get to 55 millivolts for that neuron, the whole neuron kind of changes what it does. When it reaches negative 55 millivolts, the channels that are blocking sodium and potassium, remember sodium, there's more outside, potassium, there's more inside. All of a sudden, when it reaches negative 55, ion channels start to open. They are called voltage-gated ion channels because they depend on the charge. And what basically happens is when you get to negative 55, channels start opening to sodium. So think about it. If you were sodium and you were spending your whole life outside the neuron, what would you want to do? You'd want to rush in, and that's exactly what sodium does. So when you reach negative 55, there's what's called a sodium influx. Sodium rushes into the neuron. So if you're the neuron, what do you think happens to the charge? The neuron actually turns from negative 55 all the way to positive, basically, because channels open up to sodium. After the channels open to sodium, all of a sudden they close. The ion channels close to sodium, and then they start opening to potassium. Now, if you were potassium, what do you think you want to do? You want to rush out. And that's exactly what potassium does. It rushes out. So these are some of the local structural changes that occur. So you got a sodium influx. You've got a potassium efflux, E-F-F-L-U-X, I believe is the spelling of that. So you've got a sodium influx, a potassium efflux, and then the neuron is kind of done doing its changing here. The sodium-potassium pump comes into play and actually restores the balance. Back to sodium on the outside, potassium on the inside. There's also a refractory period where a neuron can't do this again for at least another millisecond. It's called a refractory period. So these are the local events that actually occur: sodium influx, potassium efflux. The videos will hopefully show this to you a bit more. They're happening right on the surface, right at the beginning of the axon in a place called the axon hillock, right at the beginning of the axon, and they kind of go down the neuron a bit. But the last thing I have to share for you in this dense presentation is how does the signal start at the beginning of the axon and make it all the way down to the end of the axon towards the synapse. The name of this process is called saltatory conduction. And I think there's some pictures and videos to watch of this as well. Page 119 of your book. Basically, there are spots along the axon that are called the nodes of Ranvier. And basically an action potential is recreated at each one of these nodes of Ranvier. The areas between the nodes are called, there's myelin sheath, which insulates the signal, makes it go really quickly. And so basically it goes from point to point to point in this saltatory conduction fashion. I hope I've explained this to you reasonably well. If you need to re-listen to this, please feel free. Watch those videos that I've connected up to this particular part, because this part is pretty important. Finally, I just want to say, if you're wondering why are we learning all this, when we talk about drugs a little bit later, drugs exert their effect by changing this stuff with electrophysiology. So now you should understand and be able to type up a nice story for me, what happens during the resting state and what happens during the action potential. We have the local structural events with the action potential, as well as the signal transmission, which is the signal being sent down in terms of saltatory conduction. Thank you very much. Bye now.